Okay, gram-positive bacilli. Can you even name one? Got anything in mind?
C. diff.
There you go. So, there's always people trying to organize all these microbes. How do you create some order and organization and relationships? And the way they do this in this chapter, right off the bat, it's the spore formers. And I told you even in Unit 1 about endospores. I even told you bacilli and clostridium are an important group of spore formers that we're going to cover. And then within this group, when you get down, number 2 category here, this is just like your regular plain gram-positive bacilli. And then down here, there's something a little unique about these guys down here, either related to staining properties. I've already told you in lab about Mycobacterium tuberculosis and how it doesn't stain properly, and we have to use what kind of stain when it comes to that organism? Acid-fast stain. And then it becomes irregular shape for some of these others. So one or the other, irregular shape or irregular stain. So for the endospore formers, this is the ones that we'll be talking about. And again, genus Bacillus, genus Clostridium. So know that these are all spore formers, and it plays into, in part, the pathology associated with them. And there's one other thing when you look at this group that I hope that you're becoming aware of. And that is that the Clostridium group are all obligate anaerobes. The Bacillus group is not. What's happening? No.
What happened?
And some other things related to the spore formers, like you see anthracis up here, Bacillus anthracis. This is like terrorism related to the spores. And then you have C. diff associated with the spores. If I'd take a bunch of antibiotics, I'm going to wipe out everybody except the spore formers. Then I'll just have intestines just full of spores. And who do you think is going to start overgrowing? And botulism, botulinum, Clostridium botulinum, related to the spores. We had a lab that showed, hey, you might be able to kill vegetative cells, but it's going to take a bit more to kill the spores. And if you don't do that with home canning, you better watch out for botulism in your home canned products. And that's completely related to the spores. So this plays in. So we'll talk about anthrax, anthracis. Anthracis is the species, anthrax is the disease. There's some different clinical manifestations, some worse than others. I remember this whole thing. I was sitting in my TV room at home, and it was September 11th, 2001. And I had a 1-month-old on my lap watching TV. And then 9/11 happened. And then a month later, all of a sudden, there were reports of people sick and dying related to a white powder in the mail. And I'm like, what is going on now? I mean, if you were trying to protect your own back there, you're like looking over your shoulder practically. It's like, what's next? And in fact, I didn't even bring the mail in the house. I was like sorting it outside of the house. I didn't know what to do with my mail. And the mail was tainted with spores of Bacillus anthracis. That was the white powder. And if you opened the mail and were handling the mail, you were like high concentration of these spores. And if you inhaled them or ingested them, that was the worst of the possibilities, because you have pulmonary that is pretty deadly within days. Even GI is pretty dangerous. So ingestion versus inhalation. Nowadays, and this was being sent, and the thought back then was, well, geez, is it the same people that made the Twin Towers fall? And it actually was just some, what was it, like somebody from Ohio or something like that. I can't remember exactly who it was. It was just one person just kind of upset at the government or something. Like, couldn't you pick a different year to be upset? Did it have to come within 1 month of 9/11? I mean, it was a mess. And so what they've done here is they've used to have mail handling facilities on government property or within the buildings that were the people getting the mail, and now they've moved to an off-campus mail handling facilities for government mail, and they're using dogs to sniff the mail. You know, it's just what? It's just, where are we going in life? It's just crazy. One thing that's very particular on the cutaneous side, this eschar. It's a black sore. So you can see it here on this person's chin. That's a characteristic kind of thing, a black eschar. It's all pretty deadly, but some worse than others. Inhalation. Anthrax. But you know what? You'd think, okay, that's scary stuff, biological warfare kind of thing. But you know, like, you're probably stepping on Bacillus anthracis right now. You know, I mean, in the soil, it's present. You know, I mean, if we just went out and just randomly grabbed some soil and plated it out on agar plates, it'd be like one of the colonies. Which is why I don't have you go out and grab soil and plate it out. That's actually a concern. Having students plate out soil and grow up organisms on agar plate. This is the concern. So, if you're, maybe late, I would say, hey, late in the semester when you guys are like used to handling things and you stop licking the surface of your agar plates and stuff like that, throwing microbes at each other. But I don't know. I can't get you guys to stop putting the yellow bulbs in the disinfectant jars.
He was, it's the same thing. It's always happening every semester. Sounds like.
It's only getting worse. The more I bring it up, the more it happens. Yeah. The more I teach my kids how not to overflow the toilet, the more it happens. Yeah, the things you try and do to prevent. You should teach your little kids that if you feel like you're going to throw up, you should do it in the bathroom, like in the toilet, just not randomly wherever you're standing. Good luck with that one. I mean, as an adult, you learn, I think I'm getting ready to throw up. Right? You kind of know. Well, they should teach this in like kindergarten or something. Speaking of which, look at emetic toxin. Okay, so be serious. Let's be serious. Okay? And we're talking about, when you think about Bacillus cereus, think foodborne related illness.
The big scary chemicals.
And very good, it's big scary chemicals. So what do we got? We got B, S, C. What does BSC mean to us? It's big scary chemicals. What does that mean?
So serious, Staphylococcus aureus, Clostridium.
Well, right. Each organism has each letter is a name of a genus, but it's also the big scary chemicals, right? Well, what does that mean? What does that mean in terms of the type of disease? Well, it's foodborne, but that's not what I'm getting at. Talked about this in lab. I mean, it's really right on the screen. It's there are infections and there are intoxications. Okay? We were talking about this with our GI lab. And so we're not ingesting microorganisms here. We're ingesting, you cook your food properly, you kill off all the microbes. You went out and got the little leather holster that you put your thermometer in, and you walk around the house taking the temperature of everything. Make sure that everything's killed off, but yet you still get foodborne related illness because we're talking about an intoxication. Okay, big scary chemicals means intoxications. So big scary chemicals means those organisms that cause intoxications. Big scary chemicals, the chemicals are the toxin, but B in big scary chemicals stands for what? B cereus. S stands for Staphylococcus aureus. And then C's coming up. That's Clostridium botulism. So I want you to understand, you could take microbiology somewhere else, but you wouldn't get big scary chemicals. That's mine. That's trademark. This is a really good mnemonic, but you need to know your mnemonic. It's comes at you in different angles. It's very good mnemonic that way. Okay, but there's another thing. The B and the S. B cereus and Staph aureus. I want you to circle those. And I want you to draw an arrow to that circle, and I want you to say fastest onset of the ones we're talking about. And here's what I want you to be thinking about when it comes to fast onset. You went out to eat. You spent a lot of money. You paid the bill. You get in the car. You're heading back home. And then you're driving down the road and you throw up all over your dash. Okay? Like you couldn't even get home. Okay, you got this vomit everywhere in the car. When I say fast onset, I mean fast onset. Sounds good? You got that visual? Vomit all over the dash? Okay. That's what we mean. Look at this. Emetic toxin, fast onset, less than an hour. Now make that vomit noise. Blech. Okay? Think about B cereus. B. serious and Staph aureus. I had my son, we were coming back, and he threw up in the back seat of my truck. Vomit all down that little pocket behind the seat.
yeah.
You know, I had to get in there and scoop it out, and it was fun. I got a wet vac out, and I just stuck the wet vac down there and just like sucking it out of the little nooks and crannies.
I had something similar with, I was driving my brother's car one time. I didn't want to get puke, I was sick, I didn't want to get puke all over his car, so that.
Just on you instead of the car. Boy, you're a real good brother.
I called him, I was like, "You're happy with me right now."
Yeah, he still wasn't happy.
No, he wasn't.
You could just say it could have been worse. Okay, so you're getting the idea here. We're not only learning about B. serious, but we're tying it to other organisms. We've got some other mnemonics to go through. This is just one of them. Wait till we get to clevs. Okay, now we're into Clostridium. These are all obligate anaerobes. We've got a list we'll go through. I want you to be able to look at these organisms. You've got an exam coming up. I want you to be thinking diseases. What kind of diseases? What are some other unique things? Clostridium, you think obligate anaerobes. Clostridium, you think spore formers. You know, these things should just come to you, okay? You know, what if I give you a clue that it's a spore former? What if I give you a clue and it's an obligate anaerobe? What if I give you a clue, and we've already brought this up in lab, about this disease called gas gangrene? Like there's like one organism in this list causes gas gangrene. We talked about it in lab. Any ideas? Perfringens. Any idea about tetanus? Tetanus as a disease. C. diff. I think we kind of talk about that a lot. Botulism. So we got some pretty unique things to go through here. There's two things on this I want you to tie. Some of these are like when you think foodborne related illness. A lot of times you think just sitting on the toilet, you know, doing a lot of texting and Googling. Okay? Some diarrhea. But I want you to also, in your understanding of foodborne related illness, I want you to be thinking about that certain organisms are getting into your body and toxins are getting produced, and those toxins circulate through your body and cause neuromuscular problems. It's not a sit on the toilet problem, it's that you basically could be paralyzed because of this interference in the proper function of neuromuscular junctions. Well, I want you to be thinking about these two related to this. Okay? Now, these aren't, like tetanus is not a foodborne related thing, but botulism is. So you need to be able, and that's what those charts are for. Like if you're like, okay, what's the name of the organism and what body area? You're going to be in the nervous system, okay, for these things. But some of these we know as foodborne related as well. All right, so Clostridium perfringens. There are a lot of organisms that cause gangrene. Gangrene is just like super infected soft tissue. You know, you kind of like see it in the movies where somebody tells somebody else, hey, go smell that. You know? It's just like getting pretty right. That's like what they kind of like generally call gangrene. But there is a very unique category of gangrene called gas gangrene. And when you think of gas gangrene, you think of this one organism. Okay, Clostridium perfringens. And the disease is also kind of known as myonecrosis, which is basically necrosis of muscle tissue. And there are some predisposing factors like gunshot wounds. So like, small holes going deep into soft tissue. Surgical incisions, compound fractures, where like the bone might be sticking through, right? And so if this organism gets in through these wounds, then it could lead to gas gangrene. So gas gangrene is, you have necrosis of muscle tissue, but you also have swelling. So this is a thigh here, it's very swollen. A lot of necrosis going on in the muscle tissue. The organism gets in and it ferments. It ferments sugars in the connective tissue, produces gas when it ferments. So gas production. Spores get in, they germinate. Gas gangrene has a lot of different views that you can get from gas gangrene. This is actually a YouTube here. This is what's called diabetic gas gangrene. So there's an emergent treatment of gas gangrene. So it's definitely something to be aware of. For treatment?
Do you know what's actually really sad?
What?
Is that hyperbaric oxygen would really help these patients, but by the time that your insurance will cover that, it needs to be like a stage 3 wound, and they have to have like 30 days of prior traditional wound care therapy. But by the time that happens, you might as well just amputate the appendage. Super sad.
I mean, that's a pretty big hyperbaric chamber. I know they have smaller ones, probably more economical.
They have 3 of them in the basement inspector? Yeah, 3 smaller chambers.
I mean, what kind of costs are we talking about? For,
I'd say like probably at least 5,000 per one treatment.
yeah.
And you go for 2 hours for like 5 days a week for like
Mhm.
8 weeks.
Yeah.
It's like almost, yeah, they say it's like a full-time job going to these kind of treatments.
Even if they have anything.
Mm-hmm. Yeah. It's, no, it's really sad. A lot of people, like a lot of older people, like lose their legs, their toes, and
Mhm.
because it gets so bad to the point they can't do anything about it.
Related to what disease?
Like diabetic ulcers and stuff like that.
Okay. So they're doing that, not related to an infection.
I would still think it would be the same. Like,
Yeah. Well, what's an interesting thing here is using oxygen to control an obligate anaerobe. I mean, that's actually a pretty interesting thing. I mean, it's not like they're gonna like become resistant to oxygen. It's a whole different thing. But antibiotics and removal of dead tissue and other kinds of things. So that's interesting about how the insurance company dictates treatment in a lot of ways, for sure.
Yep.
I mean, you see that in a lot of areas of treatment. You know, I mean, it's an interesting thing. Okay, so when you think of be serious, did I give you some good visuals? So what would you say here? Ingestion. And think through the other things we talked about. Big scary chemicals. Tetani, tetanus. And when you think of tetanus, I want you to think of being locked up, aka lockjaw, but that's just like the jaw, but it could be arms, it could be your like a whole body where you're paralyzed, but it's locked up. And it's neuromuscular, like I was saying. This organism produces a neurotoxin. And that neurotoxin affects neuromuscular junctions. So basically, muscles can contract, but they can't relax. And so the way to think about that is just because the kind of paralysis we're going to be discussing today is a rigid paralysis, which is this, and a flaccid paralysis. You're going to see that's going to be Clostridium botulinum. Rigid versus flaccid. Tetanus versus botulism. Neonatal tetanus, rigid paralysis. So how do you treat something like that? I mean, you can go after the organism and try and kill the organism, but what we need to do is deal with a toxin, a neurotoxin. So antitoxin therapy exists for these kinds of things. But there was enough of this going around because it's like very present in soil, just like Bacillus anthracis. Clostridium tetani is just in soil. You just don't want to get the organism into like cuts and stuff. So, this is where you say, hey, tell your kids, not to run around with bare feet, or did you have your tetanus shot, or people talk about stuff like that. But anyway, there is an antitoxin that can help. Muscle relaxants. Muscle relaxants won't work for flaccid paralysis, but they would to some degree with rigid paralysis. And also a real popular vaccine we talk about is DTaP, because it covers tetanus. That's the T in there. And then some respiratory related. Somehow they decided to put it all into one package: diphtheria, tetanus, and pertussis. And diphtheria and pertussis are respiratory, but the tetanus is a neuromuscular problem. And then there's boosters. I think maybe like 2 years ago I got a booster. The booster is actually a Tdap, I think it was. Yeah. So there's a regimen of continuing vaccination for this. Okay, C. diff. Okay, we kind of call it C. diff, but C. diff is actually the name of the organism. And then you have different diseases. Enough different diseases that we call it C. diff associated disease. you'll see we have a couple different outcomes that could happen here. So one of them is this idea of diarrhea. Okay, major cause of diarrhea in hospitals, and often associated with antibiotics. Some people have C. diff as a normal resident in their colon. It's, hard to say. You could, we did some microscopy of this, and, we went around and we looked at these under the microscope, fecal material. Sometimes you can see spores. So, what organisms would cause, produce spores. So this is one of them. But it's in low numbers, and hopefully you're getting an understanding in microbiology that there are many strains of individual species, and not all the species are necessarily pathogenic. Like E. coli, we have non-pathogenic E. coli, and we have pathogenic E. coli. There's like over 150 different strains of E. coli. Well, there's multiple strains of C. diff. So what strain are we talking about? Now, there is a particular strain that produces toxins. So you have your toxigenic strains and non-toxigenic strains. Well, the worst kind of outcomes of C. diff-associated disease are going to be with the toxigenic producing strains. And you see they have what they call toxin A and B. In fact, to determine this, they would sample fecal material and do an ELISA, which you guys did in lab. They would do an ELISA for the toxin from fecal material. Okay, a big thing when it comes to caring for C. diff patients is healthcare worker hands. And so that is something we've talked about in lab related to what do you need to do with your hands when you're caring for a C. diff patient. Soap and water, not the alcohol pump, right? So this is an important thing related to what we've been talking about. So let's say that there's a patient with C. diff this patient has the toxigenic strain. Let's say I'm a person who doesn't even carry C. diff as my normal microbiota. Okay, and then I'm now, through healthcare worker, I'm getting contact, maybe through fomites, of spores from this toxigenic strain. Okay, so this can move around in hospitals in that way. So there's a lot of work to prevent that. So if you're ingesting C. diff spores of the toxigenic strain, you can get you could get diarrhea with non-toxigenic strains. But when you have toxigenic strains, we want to talk about some more severity. And one of those level of severity is called antibiotic-associated colitis. And you see down here under clinical manifestations what we call pseudomembranous colitis. So this is, might be a situation where there's just a pretty foul-smelling diarrhea. Or it could be also what we call pseudomembranous colitis. And when you think of pseudomembranous colitis, you want to think of this. This is pseudomembranous colitis. Okay, this is a normal colon. And then if these white patches develop, that's what we call pseudomembranous colitis. And it can be very painful. And perforations So if you have pseudomembranous colitis that's getting really bad, it could lead to perforations of the bowel. So these there's quite a variation of what could be and what we are calling here C. diff-associated disease. So I saw this diagram, and I thought I'd include it in our notes. It's not from your textbook. But basically, let's say that you're just some random hospital patient. You're not there because of C. diff, but you're just in a hospital. And you may have an alteration of your gut flora because of the use of antibiotics. That could lead to C. diff-associated disease. But you could also be exposed, if you're there for other reasons, that you're not even on antibiotics. You could be exposed to the toxigenic strain, say, for example, through healthcare workers' hands. Okay, so now we have a patient that has new exposure or an overgrowth of their own C. diff. So now C. diff has been acquired, but that could go nowhere and lead to a symptomatic condition. All right, so just because you're exposed to these things doesn't mean that you're going to get disease. It's the same thing with tuberculosis. And we'll talk about that, later on in this chapter, but that doesn't always lead to disease. So depending on clinical risk factors, immune status of the patient, you can get C. diff-associated disease. Okay, so diagnosis of this, as I said, ELISA, enzyme-linked immunosorbent assay for that toxin A and B in the fecal material. Mild cases, you might just focus on fluid control, taking away the antibiotics if it's a patient that had that as normal microbiota. Severe infections, getting into the use of antibiotics. For chronic conditions, what is now more and more prevalent is replacement cultures. Fecal cultures. They would find some kind of donor, analyze fecal material of the donor to make sure it's not carrying pathogens, and I'm not all sure what all the criteria they're looking for. And then when they administer that, it could be, I've seen it where it's a tube that goes through the nose, past the stomach into the intestines, and they're just pumping it in. I've seen where the use of frozen pills. Also, like instrumentation that they use, for like colonoscopies. They can also use that kind of instrumentation to administer replacement cultures. And that is becoming, in the beginning it was like, what? And now it's like, okay, here and there you start seeing, it being used and the data being analyzed about the outcome of it. Now more and more are aware of its effect and its usefulness and its success in treating people because C. diff could be a chronic condition that goes on for a long period of time. And these are the people that typically are being viewed as candidates for replacement cultures. I read a story that a mother had C. diff and they used the daughter's fecal material as a replacement culture. And they found out that, I don't know how much time went by, but the mother put on 15 pounds. and it's being tied to just a change in fecal microbes. That's interesting. Kind of makes you wonder. Okay, our last one we want to talk about is Clostridium botulinum. That's again in our big scary chemical list, so you see it listed here as an intoxication. But we're going to bring back Clostridium perfringens and talk about it again, because here we're talking about Clostridial group under the subheader food-related, food poisoning. And perfringens comes into play again. So with perfringens, it's a twofold: gas gangrene, foodborne related illness. And again, like I've been saying before, when you see botulinum, think not only intoxication, it's in the big scary chemical list, but think neuromuscular. And then also tie that back to how does that compare to tetanus. So you want to be able to compare and contrast these things. So for botulinum, there is a toxin called botulin. That is the neuromuscular junction affecting toxin. This comes up here and there, but it's more so associated with home canning. How many home canners are there here?
They're not this.
Is that right? Have you helped?
Mm-hmm. But she doesn't do like weird stuff. Like some people like canned meat.
Oh, well, canned meat could be good. I've used canned meat that I buy at the store when I dry it down. When I go in the wilderness, it dries down and rehydrates better than regular meat. And just taking and then what do you can?
I just started, but I do hot peppers, banana peppers, and
Oh, okay. Yeah. So do you use pressure cookers?
For the second batch, the first half I did, I used a stock pot and then just put towel on the bottom.
Yep. And so is that just for cleaning of the equipment, like sterilizing of the equipment at first, or?
Both.
Okay. And I want you to fill it with food. So you have the glassware, like the mason jars, and they have to be clean. And they could even be sterilized. Some people just clean them really well, and other people sterilize them before you. And how are you going to sterilize something? You know, an ideal way is using a pressure cooker, because basically a pressure cooker is going to be like an autoclave where you can get steam heat driven into spores to kill them. And so cleaning the glassware properly, and then once it's filled, then you have to heat that food up that's in those jars, and then also kill whatever may be in that food. Because if you seal it up, what you're doing is sealing it away from oxygen. If this organism is present, you just gave it a condition that it loves. You just gave it a lot of food, and you took away the oxygen. And so it's just going to have a good day. It can germinate, grow, produce that toxin. And so that's where this comes up. Even like I have a jerky maker. And, jerky you can just like slice, thin slice, and then dry, and then season it and all of that. And the jerky maker actually uses more ground meat. You can grind the meat up and mix it with some different flavors, and then it's basically extruded like a big pipette where you're pushing this plunger out and extrudes what looks like a piece of meat. But it's actually ground meat. But it actually comes with packs that contain nitrates. So you mix these nitrates in with your product, and that's supposed to prevent the growth of Clostridium. And so there's different things that are used. It's not just home canning when you're kind of concerned about this, but when you seal up cans, you're sealing it away from oxygen. So things are more standardized in industry, commercial food production, but the home canning business, that could be where the issues come in. So it's a neuromuscular toxin, and in this case, it leads to flaccid paralysis. And these can be some early symptoms. You know, what if you're like eating some food and all of a sudden, like, it could be like, you know, your mouth's not working right, you know, your half of your face feels like it's drooping. You get an eyelid that's drooping on one side of your face. Or your speech isn't right and your mouth's not like forming words properly.
But it's on both sides, not necessarily like one, like in a stroke, you see it like on one side.
No, it could be one side.
Oh, so is it?
It just depends on how much toxin is present.
Okay.
Where is it, once it gets into the system, what arteries is it getting into and affecting what muscle areas?
How fast is it, like, is the onset?
The onset for this is not very long. So this could be like within a day of eating the food.
And how long would you say, like, it would stay in this, like, your symptoms would last?
It depends on, you know, for one, the severity of it. Like, I mean, this could be deadly. If we're talking about respiratory muscles.
But do you think it's, confused with if people were presented in the ER with this, like, oh my God, you're having a stroke, versus, oh, this?
Okay, so it could, you know, when you think of something like this, you think of, like paralysis, you often think of like whole body paralysis. But what we're talking about, it could be in varying shades. So you'd be like some areas of paralysis in the body. So what could that be due to? So you think about, you know, potential meningitis, things like blurred vision could be a meningitis kind of issue, headache, blurred vision, stiff neck kind of things. But, you know, you don't get that stiff neck here, okay? But you could have difficulty swallowing because of flaccid paralysis of muscles.
There's like many other things that may be considered too, like myasthenia gravis.
Like, you think it could be that too?
Oh, yeah, yeah. Oh, yeah, well, particularly with eyelids.
Yeah.
Yeah, so you have to weigh this out, but if it's at a level that's life-threatening, you know, things have to get figured out pretty fast. Infant botulism is a thing. You know, babies are really small. You could get into what we call floppy baby. So it's flaccid paralysis, but it affects like the whole body. Because they're small and the amount of toxin that is ingested might be a small amount that might only affect like a part of an adult body, but it could affect the whole baby. They talk about not using honey, wild honey, for don't give wild honey to babies because there could be this organism present in honey. There is even cases of wound botulism, so this isn't related to food poisoning necessarily. avian botulism is a well-known thing, particularly in Michigan. Avian botulism has killed thousands of birds. And if you have ever been to Sleeping Bear Dunes, this is an area that has been well known in Michigan to have avian botulism. The problem with avian botulism is a complicated problem that is related to the mussel problem in the Great Lakes, the zebra mussels and the quagga mussels. And so if you think about our, I can get this thing to work. Why is it not working? Is this going to work? Okay, so you have the shoreline, and then you have the lake. Okay, and then they're finding dead birds up on the beach. And in some cases, hundreds of them at one time. So why? You analyze these birds, you find out they died of botulism. Now, how does this play into mussels? It's a complicated thing. So mussels initially were attaching to hard things. So like if you go offshore here in Muskegon, you're just going to have a sandy bottom going out. But if you go from Sleeping Bear Dunes and really about like Ludington north, it's going to be rocks. Okay, so these, it used to be thought these mussels would only attach to rocks, but now we've figured out they can develop communities just on sand. But we know they attach to things like boats, rocks, all kinds of stuff. So up there, offshore, there is a rocky bottom. Okay, so these are rocks. And you get the mussels that attach to these rocks. Okay? Now, throughout the entire Great Lakes system, where mussels are growing, what we've noticed over time is that the water is getting clearer and clearer and clearer. Why? Because the mussels are filter feeders. They're filtering out the small, microscopic creatures, the little invertebrates in the water. And you want those because that is part of a food web. So basically that's the lowest part of the food web, and they are filtering it out. So this water is getting clearer, and you've got these mussels growing right there. And the other thing about when mussels filter out this stuff, you know what they do? They poop. And you know what they're pooping? And what's in their poop? Phosphates. And what do you, if you want to grow some things, you like fertilize your grass, you put nitrogen. I mean, it used to be you put a lot of phosphorus down on your lawn and things like that. So what you get is concentrated phosphorus. So what happened is, now the sun, here let's draw the sun. The sun is shining deeper into the water because it's clear. And you're getting a concentration of phosphorus in certain areas. Well, now they have an algae that grows. So now you got algae growing down here. Then this algae is called Cladophora. And this algae dies every fall. And it creates muck. And we know in muck there's no oxygen. And Clostridium botulinum, the organism, is everywhere. It's literally everywhere. You know, I told you about Bacillus anthracis being like in soil. So is this organism. not in high concentrations, but so basically you created an anaerobic environment with this muck. And not only that, big storms come and they throw all of this, some of this Cladophora muck up on the beach, and the birds are going along, eating insects, and they're getting exposed to this toxin. So now you got this complicated biology that's going on, where you have the mussels making the water clear. Now Cladophora is growing because the sun penetrates farther. And there's other issues related to these mussels. These little microscopic invertebrates that they eat, these mussels, they're filtering out of the water, are food for little fish. And these little fish are food for bigger fish. And some of these bigger fish are salmon. So do you know they're not stocking the lower Great Lakes with as much salmon anymore because there's not enough food? Like what, you're just going to stock it with a bunch of salmon and then have enough food and they'll just starve? So they don't stock as many salmon anymore because of this same problem that the mussels have created. So it's a really interesting thing that we're seeing happen in the Great Lakes. So you can read more about avian botulism. So it's an interesting thing. So for treating botulism, there is also antitoxins. But if somebody shows up and it's looked like botulism due to food poisoning, there'll be people that will go and find out where the source of this is. You know, go into the house where the patient came from and try and locate the food product. You know, trying to discern whether it was home canning or did this come from a commercial food that needs to be recalled or something like that. So proper preparation and canning of foods, addition of preservatives like these nitrates that I was telling you about. Okay, related to foodborne related, we also have perfringens. And really with this, it's just basic food poisoning, diarrhea related, nothing, you know, beyond that we need to be overly discussing. So this is our spore forming group. Action of botulinum toxin. So be thinking about neuromuscular junctions, be thinking about how this relates to something else. What's the other disease? What did you say? No, I mean neuromuscular issue related disease. Tetanus. Compare and contrast tetanus with botulism. Okay, and then just a regular gram-positive bacilli that we'll talk about, Listeria monocytogenes. Not a spore former. This is foodborne related illness. And a couple unique things here. Now, this organism here, gram-positive bacilli, it can enter phagocytic cells and actually move from phagocytic cell to phagocytic cell without ever leaving a phagocytic cell. It has a special ability to do that. So that can disseminate around the body. That's a unique thing about this organism, and it causes a disease, listeriosis. And this is foodborne related. Often associated with dairy products. So if you had to put your finger on this organism and what kind of foodborne food sources, more so it's associated with dairy products. So things like milk, cheese, ice cream, yogurt, and but it doesn't just stop there. There was a case, as you can see here, in 2011. 35 people in Colorado died from cantaloupes, contaminated cantaloupes. Okay, so there can be some other food products other than dairy. But this is also one of these reasons why because of its prevalence, pregnant women are often advised not to eat certain foods. And some of those certain foods are unpasteurized cheeses. Okay, so cheeses like, I don't have in my mind a complete list of unpasteurized brie. And what else?
I think you have hot dogs and bologna.
Hot dogs and
Yeah,
bologna.
it's like deli meat. Some
Deli
of the
meat.
Soft cheeses.
Well, the soft cheeses are often the unpasteurized. Okay, so we know this is foodborne related. We know a little bit about its virulence factors, about the living inside phagocytic cells and moving between phagocytic cells. What makes it so deadly? And this is not just a foodborne intestinal issue. This organism can spread beyond the intestines. And this is where it becomes a very dangerous organism, because of that ability. Can spread to meninges, get into cerebral spinal fluid, different kind of organs, blood infections. These are the things that are killing people, not the diarrhea, but those kind of invasive issues. And the other thing, related to pregnancies is this organism can cross the placenta. So that is a very important aspect. So there's been a lot of recalls that you hear about with Listeria. I remember there was a ice cream manufacturer in Texas. I forget the name of it. They recalled all their ice cream, and it just comes up quite often. Food recalls related to this organism. Okay, so think about, some of these organisms that we're learning about cause foodborne related diarrhea, and some of them go beyond. We talked about one that goes to neuromuscular, and now we're talking about one that has another kind of dissemination. Could also be neurologic. Okay, and so in our last group, we want to talk about these irregulars, whether it be due to them being pleomorphic or some other kind of staining issue. So we'll talk about, even though we know they're gram-positive bacilli, they're irregular in some way. So we have Corynebacterium diphtheriae. This is generally bacilli, but you can see there's some pleomorphic nature to it. So under the microscope, it can be very difficult to characterize its morphology due to this. Causes diphtheria. So diphtheria, is this a lower respiratory tract infection or upper? It's an upper respiratory tract, so we can abbreviate that URT. So like in your charts, as you fill them out, you can write URT. And then as you're learning about bacterial diseases of the respiratory system, you can organize it into URTs and LRTs, lower versus upper. So this is one of our upper. And it forms a leathery pseudomembrane that in some cases can be observed, and in some cases it's deeper, that you can't see it, just by observing through the mouth. This membrane can get big and bigger and bigger, and it can actually block the airway and lead to asphyxiation. So associate this leathery pseudomembrane that can get bigger like that with diphtheria. Associate that vaccine with diphtheria. A lot of deaths before that vaccine. It's a really interesting thing to see in a community a certain level of diseases, and then you have the vaccine administered and you see that drop off. It's really pretty impressive. So that's built into that DTaP. Next one is Propionibacterium acnes. The disease we're talking about here is, of course, skin related. It's acne. So that's really it. Not much to learn there. It's like right in the name. But kind of a fun, this is like what you talk about at parties. Okay, microbiology. How do you talk to people you don't know at a party? Do you say, nice weather we're having? Or do you say, did you know that Propionibacterium cause acne, and they also give sweat its odor? So you could say that at the party. That could be like the initial line. And then people will be like, wow, that's interesting. And then you tell them, did you know that it also is like the flavor of Swiss cheese? They use Propionibacterium to make Swiss cheese. And it's the same chemical that gives it its flavor.
It's dry.
You see, this is important conversation-making stuff. And I think, maybe what we could do in micro labs of the future is actually collect sweat and see if we could make some Swiss cheese out of it. What do you think? Is that a doable thing? Okay, the last one here is Mycobacterium. And we're really only talking about Mycobacterium tuberculosis. This is something we've been discussing in lab with our respiratory lab. We're getting into talking about tuberculosis, which is like the #1 disease in the world caused by a single pathogen. We mentioned that in Unit 1. I want you to understand that there is a waxy substance called mycolic acids that's in the membrane. And that waxy substance will not allow the gram stain to gram stain it properly. We know structurally that this organism is a gram positive. So it has that thick peptidoglycan layer, but that membrane is filled with, is intermixed with mycolic acids. So, but it doesn't allow it to be stained properly. So you use acid-fast staining to work with this. And we talked about that in lab. If you're trying to rule in or rule out that a patient has or doesn't have tuberculosis, and you get sputum sample, what staining technique will you go to? You don't gram stain it. You will acid-fast stain it. Okay? That's an important thing to know. And on agar, if you can just kind of imagine this bacterial colony right here and put your finger down in it, it's waxy because of this. And it's also like super slow growing. Okay, so this causes tuberculosis, and we'll get into the pathology of that. These are the individual little bacteria. So these are all little, they're all little bacilli here, little bacilli all over in here. But then in some times they all stick together into what is called a cord. Okay, so sometimes you see that. This is not one giant bacteria. That is hundreds of thousands of bacteria that are kind of stuck together. And this kind of plays into the pathology of what happens in the lungs, which we'll talk about. You know, the fact that this waxiness prevents phagocytosis and destruction. So that's a virulence factor in itself. All right, so tuberculosis is, like I said before, it's the #1 disease in the world by a single pathogen. We learned about that in Unit 1, but not so much in this country. And the reason for this is related to nutrition. So in other developing nations, there are issues with inadequate nutrition and access to medical care. These are big players. So these are big predisposing factors in a lot of countries. Okay, it's spread in respiratory droplets. And the diseases we want to talk about is tuberculosis as a whole, but I want you to understand some depth to that. So we want to talk about what is primary tuberculosis? What is secondary tuberculosis? What is disseminated tuberculosis? And the other thing I want you to see here, when it talks to talks about the number of people. Let's read this. Only 5 to 10% of infected people develop clinical disease. Okay? Let's say you're caring for a tuberculosis patient. You're taking respiratory precautions. But let's say something happens and you're exposed. Okay? So we'll go through and we want to see that that's a pretty low proportion here. Okay, that would actually develop clinical tuberculosis. So what is primary TB? Primary TB is characterized as a disease of the lungs where tubercles are formed in the lungs. And it could be hundreds and hundreds of them. Here is these tubercles that you see here. There's even this kind of an issue with deer in Michigan. This is one of the diseases that you're always on the lookout for when you get a deer. And you open it up and you're pulling out all of the gut content and cutting open. You cut through the diaphragm away from the exterior cavity, and then you start pulling this lung material out, lungs and esophagus and all of this, and then you can start to see whether or not you see tubercles. Sometimes it can be on the outer walls, but they are a tubercle, and if you were to do scans of these, This is a cartoon of what a scan would show and what we call granulomatous lesions. And so what we see here is you have the bacteria present here, but overall what this lesion is really only a small part bacteria, and the rest of it is actually all of the immune system and lines of defense coming in to fight this bacteria, and they are causing the tubercle. They are causing this granulomatous lesion because you have, within here, you have all kinds of cells that are T cells and B cells and neutrophils and all kinds of other cells that are coming in to fight this. So you can see this on scans, and I thought I had somewhere. Let's take a look here for a second. I don't have a, I thought I had a lung scan in there. Well, not a lung scan, but a, like a histology. So over time, if you were to let's say you had this stage of primary TB, and some lung sample was collected, and there was some histology done. And let's say you're cutting fine slices through these tubercles. And as you look at it under the microscope, you see a lot of cellular detail. And you can see the types of cells that are present, if you're good at identifying cells. Over time, this structure will go into a stage that we call caseous lesions. Caseous is like milk, and what it is where it becomes to a point where it's gone necrotic, where when you finally slice through it, you don't see that cellular detail anymore. All you see is like, if you were to slice through a thin slice of cheese, all you see is just no particular cellular detail. So you can see where this is what they call caseous necrosis, and then that will eventually be the whole thing. So sometimes these tubercles will have cellular detail, and then later in the stage they'll go caseous. That's primary TB. Secondary TB is where these tubercles expand, and now they develop into a more advanced respiratory disease. So this is where you get fluid buildup in the lungs. You get extensive and violent coughing. The type of sputum can sometimes have color to it, not in all cases. Greenish in some cases can be an indicator. Bloody sputum, and can lead to a host of other issues, and can be fatal in itself. Okay, so primary TB is one thing. Secondary TB is definitely a more advanced issue. And then disseminated TB is sometimes referred to as extrapulmonary TB. And this is where it can spread from the lungs to all areas of the body. Okay, so you've got these three types of TB that to know about. And this table is there. You can kind of look at some of the detail in there, but really the one factor that I wanted you to know about was related to what was being said right here. So if TB bacteria are inhaled into the lungs, this may lead to no disease and statistically, you're only talking about 5 to 10% would actually lead to disease. So that's the only factor I gave you earlier. All right, so you can be thinking about this. There's the TB skin test. So this is where you use tuberculosis antigens and they inject them under the skin and they look at the nodule that forms. And you have to be, you're not just looking for a nodule, it has to be a certain size, it has to be a certain color and shape. And so it takes somebody who's trained to look at that. But then also x-rays and CT scans, collecting sputum, doing acid fast staining as ways to diagnose this kind of problem. But the TB test is typically a go-to here. Treatment usually is a long-term regimen. There are maybe, I've forgotten the exact number, maybe around 8 approved drugs in the United States. There is a 1-pill regimen that actually is a combo of different drugs. Vaccine, because we're not like a high proportion of the TB cases in this country. You don't see vaccination as a big player, but in other countries it's a different story. Particularly like if you have issues with nutrition, you have issues with access to health care, this is where it becomes important. Okay, so I think we're good. Respiratory droplets.